of genetic dominance. So given that they have this genetic dominance, next slide please, one of the ideas for eyes on therapies for them is that they have a very different sort of take on what would help to cure these diseases compared to recessive diseases. And so if you think about recessive disease, many of those diseases are loss of function mutations. And the general strategy used for those is to put back the missing protein, rescue the activity of the mutant gene. And genetic and pharmacologic strategies really target that idea of replacement. In contrast, dominant diseases tend to be gain of function or dominant negative mutations. And because of that, rather than putting something back, you need to take it away. And so the strategies we'll hear today are many different ways of genetically or pharmacologically inhibiting or removing that mutant protein. Next slide, please. So today we again, we'll talk through all of these various talks. Each presenter has 15 minutes to walk us through their various strategies in these very different diseases. And so the first person to talk today will be Ramesh Subramanian, again from Dyne Therapeutics, and he is going to talk about their strategies for myotonic dystrophy. Welcome, Ramesh.
Great, thanks, Beth. Good afternoon, everyone. Hopefully you can hear me. Thank you to the MDA for the opportunity to present Dyne's compelling preclinical data in myotonic dystrophy. Just wanted to also mention that I'm presenting, of course, on behalf of the entire Dyne team who has done some phenomenal work in the 3 years that we've been involved in myotonic dystrophy, and we look forward to moving these medicines into clinic.
Ramesh, unmute. Thank you.
Apologies. So just before I begin, a reminder that I'll be making certain forward-looking statements in my presentation that involve certain risks and uncertainties. We are laser focused at Dyne on our mission of delivering life-transforming therapies for patients with serious muscle diseases. And our platform, let me advance, it's called the FORCE platform, and it's enabling modern oligotherapeutics for muscle disease. And it consists of 3 parts. If I could get this. First is the antibody. We're using a fragment antibody that targets TFR1. So this essentially directs the medicine towards the muscle. Next part is a linker. So this is a clinically validated linker that connects the antibody to our payload. And last is the payload. And since our therapeutic is modular, the antibody and link always stays the same, and we can switch out payloads depending on the target. So in the case of DM1, which is an autosomal dominant neuromuscular disease, the problem lies in the nucleus, and therefore we utilize a single-stranded antisense oligo to address toxic RNA within the nucleus. If there's a target in the cytoplasm, we would use an siRNA, of course. So this platform allows us to generate a robust portfolio of diseases. We're working on myotonic dystrophy as well as DMD and FSHD. So let me walk you through some of the aspects in myotonic dystrophy and our data. So as you all know, DM1 or myotonic dystrophy is autosomal dominant, caused by an expansion of triplet repeats in one of the alleles, and it results in a shortened life expectancy. Patients die in their 50s. And the primary presentation is a muscle phenotype. So you have myotonia, muscle weakness, cardiac arrhythmias, and pulmonary abnormalities. They also have CNS defects. Large population of patients, and currently there are no approved therapies. Our approach is a disease-modifying nuclear DMPK knockdown. And by targeting the toxic DNA or RNA within the nucleus, we aim to potentially stop or reverse disease progression in myotonic dystrophy. So let me walk you through the genetic basis of disease. So you may be all familiar with this, but I want to touch upon 2 items. FORCE targets the genetic basis of myotonic dystrophy, As I mentioned earlier, it's DNA triplet repeats that are converted to toxic RNA repeats. These RNA repeats form these huge three-dimensional structures, here represented by red as hairpin loops, and they are stuck in the nucleus. And as they are large hairpin loops, they bind a number of splice proteins, and this results in a spliceopathy, or a splice-related disease. And you can actually stain for these hairpin loops within the nucleus, and you see them as clusters of foci. Once these splice proteins are bound by these loops, it results in mis-splicing of downstream RNAs, such as chloride channels, calcium channels, as well as muscle structural and functional proteins, for example, BIN1. And ultimately, that results in the clinical sequelae, which are myotonia, muscle weakness, and particularly cardiac and pulmonary defects. So the problem lies in the nucleus, and we use the FORCE platform to deliver our molecules into the nucleus to address those toxic RNAs. So here I'm demonstrating that FORCE targets toxic nuclear DMPK RNA, reduces the nuclear foci, and corrects splicing in DM1 patient cells. So these are DM1 patient myotubes, and here we're looking at knockdown of DMPK RNA. The y-axis is DMPK expression. It's at 100% with a control treatment, which is PBS. With a single dose of our molecule, we are reducing the amount of toxic RNA by about 60%. Moreover, as you stain for those repeats that I mentioned earlier, these are shown here. Blue are the nuclei, and red are those foci. As you see, all the toxic RNA is stuck within the nucleus, and very little, if any, is seen in the cytoplasm. So we have to get to the nucleus. Now, a single dose of our molecule is shown in this picture, and you see there's a substantial reduction of the amount of toxic RNA foci within the nucleus, those little red dots. And ultimately, reduction of this toxic RNA results in a correction of splicing. This is key for patients. So on the y-axis is percent exon 11 inclusion in one of those structural proteins I mentioned earlier, BIN1. This is control. You see only 10% exon 11 inclusion. But a single dose of our molecule increases the amount of exon 11 included in BIN1 to about 30%. Tells us we're delivering a molecule via the TFR1 receptor. The payload is getting into the nucleus and degrading toxic RNA, and that results in a correction of splicing. So we've advanced our molecules into two different in vivo models, and here I'm demonstrating that FORCE dose-dependently corrected splicing and reversed myotonia in the HSALR DM1 mouse model. So the HSALR mouse model is a well-known model. It replicates some of the splice disorders seen in patients, and it does express one of the phenotypes that are seen in patients, which is myotonia. So the left-hand side, let me walk you through that slowly. It's looking at splice correction in multiple muscle types. The y-axis is splice derangement, and x-axis is treatments. And the green bar here at the bottom represents wild-type splicing. So red is a cohort of PBS or control mice showing high amount of splice derangement. Green is Dyne's molecule at 10 milligrams per kilogram, and blue is at 20 milligrams per kilogram. All these studies are done with the entire conjugate, antibody linker, as well as the payload. You see a dose-dependent correction of splicing towards wild type. And this, I remind you, is again with a single dose of our molecule. And you see that in 3 different muscle types. Most importantly, it's not a single time point. What each of those dots represents is a composite splice score taken from 35 different RNAs in vivo. So we essentially inject our conjugate intravenously, and then we harvest muscles 2 weeks later and look at splicing of various RNAs, like I mentioned earlier, the chloride channel, calcium channel, BIN1, and other RNAs by PCR. And here you're seeing significant changes in splicing, for example, here, and also on the chloride channel. And as you see with the chloride channel, we get almost a wild type at 20 milligrams per kilogram dose, single dose, and that results in the profound reversal of myotonia seen on the right-hand side. So here you see complete myotonia reversal in 14 days with a single dose. Again, let me walk you through this part. Y-axis is myotonia grade measured by electromyography, and x-axis is various treatments. PBS, you see a grade 3, which is a high amount of myotonia and greater than 50% of the muscles. Red is a free naked ASO. You see slight reduction in myotonia. And in blue is the Dyne molecule with a single dose, almost completely reversing myotonia in this model system. Tells us that not only is that splice change important, but it results in change in phenotype as well. And just as comparison, others are published in this model wherein they required anywhere from 4 to 8 doses over a month to show reduction in myotonia. So we advanced our molecules into another model called the HTFR DMSXL model. This is an innovative model developed by Dyne to evaluate the pharmacodynamics by measuring toxic human nuclear DMPK RNA. So let me walk you through the slide, the way we generated these mice. So these are mice that we generated in-house expressing the human transferrin 1 receptor. It's knocked into the mouse transferrin receptor locus. So we can use our clinical molecules in these mice. We crossed it with a well-known model that we obtained from Inserum called DMSXL. We generated these mice that express both human receptor as well as the human target. And it has over 1,000 repeats, so it represents quite a significant phenotype. Now, the key point is that all the toxic RNA is stuck in the nucleus in this mouse, very similar to what we see in patients. So to go to the data, our human force conjugate demonstrated robust toxic human DMPK knockdown in this mouse model system. So we are targeting anywhere between 30 to 50% decrease of this toxic RNA in the mouse. And what I'm showing you here, y-axis is DMPK expression. In 4 separate muscles, we see that we are in that range where we believe we'll be clinically relevant. In tibialis, we're showing a 54% knockdown, gastroc 39%, and very excitingly, in the heart and diaphragm, we are close to 60%. Tells us we are, by IV dose, the transferrin 1 antibody is getting us to the muscle and delivering our payload in the nucleus where it's degrading that toxic RNA. Subsequently, we advanced our molecules into primates. Now, primates, there are no DM1 models. So all primates are wild type. Therefore, there are no toxic RNA stuck within the nucleus. Most of the RNA is within the cytoplasm, and it's not toxic RNA, it's just wild type RNA. We utilize primates to understand delivery to muscles and also tolerability of our medicines. Here we're showing you that force achieved enhanced distribution and wild type DMPK RNA knockdown in NHP skeletal, cardiac, and smooth muscle cells. When you look at tolerability, our force medicines are well tolerated in our primate studies. All CBC measures, including iron homeostasis and platelet counts, were within the normal range. And when we assessed kidney and liver function and body weight, they were all within normal range as well. So we're very happy with this tolerability profile as well as we head towards our clinic. So just to summarize what I've shown you today, preclinically, we have targeted the toxic DMPK RNA in the nucleus for both patient cells as well as disease models. We've reduced those nuclear foci, we've corrected splicing changes, reversed myotonia, and also delivered to large muscles of both mice and primates. And I haven't shown it to you here, but we do have durability with a single dose that goes out to 12 weeks. So as we think about our dosing, we are heading towards hopefully a monthly dosing, a less frequent dosing. And all of this together gives us a tractable development plan to a rapid human proof of concept. We're advancing our myotonic dystrophy program into clinic soon. It will be one of the 3 INDs planned between Q4 2021 and Q4 2022. The team is working hard to get there. That's a picture of the team when we could get together last February, Rare Disease Day, when we actually could be in the same room. And we look forward to advancing medicines for this